Alrighty, folks. So welcome to day 2. Last time we didn't do too much. Talked about myself for a little while. Got into a good portion of chapter 1 where we talked about what is this class, why is it so fabulous, and then we talked for a while about how we classify living things. And the last thing we talked about was that thanks to Carl Woese and George Fox, we now classify all living things into 3 domains. Right? If you're alive, you are either in domain bacteria, domain archaea, or domain eukarya. And what I want to delve into here today to start is what is the difference between these 3 domains, right? What does it mean to be a bacteria? What does it mean to be an archaean? So, the defining characteristics of bacteria, okay? And these are important because we focus on bacteria as our model organism this semester. So first things first, bacteria and technically archaeans are what we call prokaryotes, okay? And the prokaryotic cell differs from a eukaryote, like humans, in that prokaryotes are all single-celled, okay? So when it comes to bacteria and archaeans, they're all single-celled, okay? Unicellular, no multicellular bacteria, sorry. But the real defining characteristic is that prokaryotes, like bacteria, lack membrane-bound organelles, okay? So this is where I make one of those assumptions you've had in biology. When you take Gen Bio, it's a big chapter that talks about all the organelles within the eukaryotic cell, okay? Human cells have nuclei. We have endoplasmic reticulum, Golgi bodies, vesicles, okay? All those membrane-bound guys. Prokaryotes, like bacteria, don't have those. No organelles, okay? You cut a bacterial cell in half, you look inside, it's a big bag of soup, okay? So they don't have that same level of organization. So prokaryotes, single-celled, no membrane-bound organelles, and they all reproduce asexually. Okay, only eukaryotes can reproduce sexually. All right, so what do bacteria look like? Well, they got 3 basic shapes. 99% of them are either bacilli, cocci, or spirillum. Okay, so what is that? Well, bacilli means rod-shaped bacteria. Okay, roughly rod-shaped. Don't think like a perfect squared-off rod. E. coli, for instance, is a bacilli. It's rod-shaped. Cocci or coccus means spherical, and these guys tend to be perfect little circles. An example would be Staphylococcus aureus or Staphylococcus epidermidis, which I mentioned in lab this morning, and I'll mention later for the other folks. So these guys tend to be perfect little spheres. And then the odd group, which we won't really get to see this semester much, are the spirillum or spirilla. These guys are curved rods. They can be just kind of a few curves, or they can be great big corkscrews. It all depends. But when it comes to shape, that's it. Okay, with a couple of rare exceptions, bacteria are either bacilli, cocci, or spirilla. Okay? Rod-shaped, spherical, or curved rods. You're going to want to know those. But we'll get into shape and what we call arrangement more in Chapter 3 when we look at cell structure. All right, so the next thing I'm going to mention, this term is exceedingly important. And it is a term that you will never forget after you leave this class. Years from now, you'll know what it means. Peptidoglycan. Okay, and I mentioned, my micro teacher told me the same thing, and she was really right. Peptidoglycan is a chemical unique to bacteria. Okay, peptidoglycan is what bacterial cell walls are composed of. Okay, so like plants, like fungi, bacteria have tough cell walls. Okay, and that cell wall is made up of, this is the 4th time I'm saying it now, peptidoglycan. Okay, you may not remember it right after today, but we will talk about this term so much in the first half of the semester, you will never forget. Okay, it's going to be really important. So they come in three shapes. They're prokaryotic. They've got cell walls made up of peptidoglycan. And now, next characteristic is membrane structure. I have wonderful news. If you've had biology before, you've talked about membranes, you've talked about phospholipid bilayers, right? Membranes are supposed to be two opposing layers of lipids. Good news. The membranes of bacteria are almost the same as the membranes of, say, humans. Okay? They've got phospholipid bilayers, we've got phospholipid bilayers. So our membrane structure is very similar. Now, reproduction, probably the simplest thing out of all of it. Okay? Reproduction to bacteria is called binary fission. Purely asexual. And this is it. Okay, ready? One cell divides itself, becoming two clones. Those two divide, becoming four. Those four become 8, 16, 32, 64, 128, and so on. This is self-cloning. It's a very straightforward process. So if you're haunted by memories of mitosis and meiosis and all the phases, I have good news. You don't worry about that here. Okay? Binary fission is ridiculously simple. Now, this isn't too bad, right? These are pretty easy defining characteristics to remember. But where bacteria kind of really turn it to 11 and get very complicated is unfortunately metabolism. It's everybody's least favorite chapter, and it doesn't get any better here. Bacteria have ridiculous diversity. And a big part of that diversity is they can eat damn near anything, which means they have very complicated metabolic pathways. So they can derive energy from chemical sources, inorganic, organic, you name it. And to throw a wrinkle into the mix, there's plenty of them that are photosynthetic. They can use the sun for energy, just like plants. So it's a lot of diversity here. All right, but any questions about what makes a bacteria a bacteria? Okay, so you've heard of bacteria before, but what many of you haven't heard of coming in here is the pore archaeans. Okay, like we talked about last time with Wose and Fox, we thought for a long time that bacteria and archaeans were the same, and they're not. Archaeans look like bacteria. They behave like bacteria. But as Wose and Fox found out, when you look at their genetics, they are distinct. They're their own group. Archaeans actually deviated from eukaryotes. So from a genetic standpoint, they're a little bit more closely related to a eukaryote than bacteria. All right, so some other defining characteristics about these bacteria-esque organisms. They do have cell walls, like bacteria. But it's not peptidoglycan. It is very cleverly named pseudopeptidoglycan. They typically have the same shapes as bacteria. Tend to be bacilli, cocci, spirilla. But some of them can take on some really weird shapes, which I'll show you when we get to chapter 3. Another interesting characteristic about archaeans. There's a reason why you don't hear about them much. They tend to not like the environments we do. They tend to like the environments nobody else does. They're often called extremophiles. They like horrible environments like acid lakes, soda lakes, boiling water near volcanic vents at the bottom of the ocean, okay, anaerobic bogs. So some examples here. So Wose and Fox studied methanogens, which they thought were bacteria. These guys are found in anaerobic bogs. This is probably in Scotland somewhere. But these bogs are really interesting. There's not a lot of organisms in bogs that can decompose anything because of the lack of oxygen in the water. And in Scotland, for instance, you'll see pictures where like sheep will wander into these bogs, they'll fall in, and they won't ever decay. They'll sit there kind of preserved forever. Lovely bright pink soda lake somewhere. This one's actually very, very salty and fairly alkaline. The bright pink color is Archaean growth. And a beautiful overhead shot of a geyser in Yellowstone. Probably really, really hot, but the vibrant colors come from not just minerals, but also from Archean growth. Okay, so they tend to like ridiculous environments. So that makes you wonder, how can they survive? They're tough. Besides having really tough cell walls, their membrane structure gets really unique. Okay, and this is something we won't get into much, and you take like 400 level classes for this, but they don't tend to have lipid bilayers. Okay, most membranes are 2 layers. These guys make a single layer, and that actually allows them to be even tougher and survive in ridiculous environments. And another strange characteristic, another reason you don't hear about them, they don't cause any trouble. None known are pathogenic. None of them are associated with disease. Okay, so hopefully you folks remember the poor Archaeans. I will mention them from time to time, but again, they behave and look like bacteria, but from a genetic standpoint, they are their own domain, their own group. All right, so again, any questions about the bacteria or the Archaeans? Okay. Now, the rest of the examples here I have are mostly just kind of footnotes because there are microorganisms found in the eukaryotic side of things. Okay, but we won't talk about them as much because they get very distinct, and you tend to have to take classes on each one individually to really understand them. So eukaryotes, again, membrane-bound organelles like humans. Fungi. Okay, say fungi, say fungus, people think mushrooms or molds. And you're right. But most fungi actually start off as being microscopic, or they stay microscopic. Like yeast, for instance. Right? We bake with yeast, we brew with yeast. Yeast are microscopic. So they can be single-celled or multicellular. They can switch back and forth. They do have cell walls made of delicious, digestible chitin. And fungi, their niche in our ecosystems is that of decomposition, right? You see a dead tree in the woods, probably covered in fungi, slowly breaking it down. And that's a really important job because there was a time on this planet when nothing could break down trees. So that is called the Carboniferous era. There was a time on this planet when it was dominated by trees, because there was nothing that could break down the trees when they're alive, and there was nothing that could break them down when they were dead. So imagine forests are just covered in these huge, dead, fallen over trees. Well, that's where the vast majority of our coal comes from, right? You dig deep in the ground, you go through the different strata, and then you hit coal. That's the Carboniferous era from all those trees. But that strata ends. The coal ends. What happened? Fungi happened. Okay? Evolution and adaptation occurred. And fungi slowly evolved what's called cellulases. Enzymes that can break down the tough, woody material of trees. Lignocellulose, whereas nothing else could. So, that's why coal's finite. And you don't have no Carboniferous era. Don't even really talk about that in Gen Bio. That's more geology. We mention them from time to time because, yes, many of them are pathogenic. Well, some are pathogenic. So typically you think, ringworm, athlete's foot, thrush, vaginal infections. They become systemic. You can have some problems. A good friend of mine died 2 years ago from fungal pneumonia. Okay, so they can be quite problematic. All right, algae, I'll barely touch. Algae belong more in a botany class, but when you think algae, think microscopic plant, because that's what they're most similar to. These are also eukaryotes. They tend to be single-celled, but they can be multicellular. Like plants, they got cellulose cell walls, so they're tough. Like plants, they're capable of photosynthesis, right? So they derive energy from the sun to turn carbon dioxide into sugar, and produce oxygen. So algae have a very important role on this planet of providing you most of your breathable oxygen. right? Trees, they do a great job. They produce a lot of oxygen. But you got to remember, about 70% of this planet's covered in ocean. And the ocean's got a lot of algae in it, okay? So a lot of your oxygen comes from those marine algae. Protozoans are my least favorite group, okay, for a number of reasons. These guys are eukaryotic. They are typically single-celled, and they're itty-bitty hunters. Okay, most protozoans are tiny hunters of other protozoans, of bacteria, and some of them, unfortunately, are parasites of, say, humans or other animals. So these guys are a mess. Protozoans are a super hodgepodge group. Okay, and classically, the only way we classified them was based on movement. So I bet you've all seen paramecium at some point. Okay, paramecium are covered in those hair-like cilia. Okay, we classify them because they use cilia to move. You've probably seen Euglena before. Okay, Euglena have long flagella. Okay, different type of movement. And you've probably seen amoeba. Okay, amoeba have no cilia. They have no flagella. They just drag themselves along. Okay, so that was how we classified them. How do they move? The other issue with protozoans, especially parasites, and why I dread them, if you take parasitology, have fun, because it's an entire semester of life cycles. Every parasite has its own unique life cycle. And that's why they're an absolute mess. And I'll mention them here or there this semester, but they're an awful complicated group. All right, so something we will talk about at some length, because we have a whole chapter on them, is viruses. And I find talking about viruses is a lot of fun because there's so many misconceptions. People just don't understand viruses well. So, for instance, the first week of any biology class, I guarantee you go through a lecture or an exercise where you have to define what does it mean to be alive, right? What is the criteria of being a cell? So you gotta have a membrane, you have to have DNA, you have to maintain homeostasis. But there's one thing that's really important that all cells have to be able to do that viruses can't. Does anybody remember that factoid from Gen Bio? What can't viruses do that, say, we can or bacteria can? Starts with an R. Yeah, there we go. I didn't have to say ends with an E, produce. Yeah, viruses don't reproduce. There's no sexual reproduction. There's no asexual reproduction. But the thing is viruses somehow make more of themselves, right? They spread. That's where you come in. Viruses lack the machinery to make more of themselves. But they take advantage of your cellular machinery to do that. So they're what we call obligate parasites. They need living cells to do the work of reproduction. So when you have a viral infection, they're getting into your cells. And they're doing one thing. They're turning your cells into factories. Make more viruses until the cell dies. So with that in mind, it would make you think that they're probably complicated, right? In order to take over cells and turn your cells into factory. They're not. Viruses are the image of simplicity. They have two major components. They have a protein shell and a nucleic acid core, DNA or RNA. So let's illustrate that. Let's all draw the flu. Okay? Anybody can draw the flu. So let's draw a hexagon. All right, organic chem didn't fail me. So that's your protein shell. Now inside of that hexagon, draw 8 lines. 2, 3, 4, 5, 6, 7, 8. You just drew the flu. Good job. I mean, there's not much to it. So you got your protein coat. Okay, that's the shell here. And then inside of it, that's your DNA or RNA. So it's a shell surrounding a nucleic acid core. Now, what's up with those 8 lines? Well, the flu is made up of 8 segments of RNA. Okay, so they're simple little things. Okay, it's usually just a shell surrounding a nucleic acid core. But I digress. We will get into that way more when we get to chapter, I think, 6. I'm still getting used to the new chapter numbers. All right, so the point here with all this is that the microbial world encompasses all 3 domains of life, right? So bacteria, archaeans, even eukaryotes can be microscopic. But in this class, you also have to regard that there are non-living agents out there, non-living particles like viruses. And then viroids and prions are stranger still, but we'll get to them more when we get to the virus chapter later on. All right, any questions about the eukaryotes? All right, so the rest of the time here, we should have plenty of time, I hope, is discussing some of the historical milestones in microbiology, because I always love this sort of stuff, so I get to instill it upon you. Sorry. And the other thing people wonder whenever anybody talks about history is, do I have to know the dates? No. Okay, not a history class. Okay, so let's start from the beginning. Let's ask ourselves, who saw the first microorganisms, right? Somebody had to have identified that they were there. It happened some time ago. Okay, late 1600s. Antonio von Leeuwenhoek was the first person to observe microorganisms. Okay, so how did he do that? Well, in the 1600s, there were microscopes. Some of them actually pretty nice, but his was fairly crude. So you got this brass plate here. You got a small but powerful lens. And then this is a sampling needle. So he hypothesized that there may be tiny organisms that you can't see without a lens. So he went out and he took samples from flowers or other parts of the environment, and he held that brass plate up to his eye. And what did he see? Okay, in the middle there, those are his pencil sketches. Okay, he probably saw pollen, he probably saw mold, maybe eukaryotes. He did not see bacteria. Okay, too small. They'll come later. So we did not call these microorganisms to start. Okay, von Leeuwenhoek had a keen sense of humor, and he said, let's call them animalcules. Right? Animal molecules, tiny molecules. Okay, but anyways, and his writings are hilarious because he sounds like an excited kid. Nobody writes like that anymore. So like I said, there were microscopes around this time, and I mentioned some of them were actually much nicer. So typically in Gen Bio, they talk to you about Robert Hooke. Okay, Robert Hooke studied plants, and he worked in collaboration with a microscope manufacturer named Christopher Koch to develop one of the first compound microscopes. Okay, so the microscopes we use in our lab are compound, meaning there's more than one lens. You have 2 lenses in order. So we do that because when you put lenses in series, their power is multiplicative. Okay, so in that beautiful mahogany gilded microscope that nobody would ever use right now, there are multiple lenses to help you magnify objects even more than a single lens would. So he wanted a nice microscope because he wanted to see what were plants made of. What was holding together leaves? So he took a cross-section of what I think is a locust leaf, put it under the scope, and that is one of his sketches up there. And he's like, whoa, it's made up of all sorts of little chambers. Okay, and he thought those chambers looked like prison cells. So Robert Hooke is the person who coined the term cell. And this hypothesis the hypothesis he formed was that, okay, let's see what else is made up of cells. And he found out everything alive is. So that led to cell theory. Now, there are no laws in biology, but there's a handful of theories. Cell theory is the important idea that all living things are made up of cells, be it 1 or 20 billion. If you're alive, you are a cell, or at least one cell. Okay, so with this stuff in mind, this leads me into the next story and milestone. At the time of von Leeuwenhoek and Hooke, there were two schools of thought as to how living things arose. Okay? Today, we know that all living things come from other living things, right? We reproduce. Bacteria reproduce. Archaeans reproduce. Okay? Organisms reproduce to make more organisms. That is what we call biogenesis. Life makes life. Some people believed biogenesis was true, but many people also believed in, and this one's fun, abiogenesis, also known as spontaneous generation, which is a little bit more on the nose. This was the idea that living things can arise from non-living or dead matter. For example, and I'll lean on this a bit here, if you leave a piece of meat out, it rots, and what appears in short order? Maggots. So like, okay, dead meat, air, and time equals the spontaneous appearance of maggots. Folks, we know that's utter crap, right? The flies land on it, they lay eggs, and the eggs hatch. They didn't know that. Okay? Another great experiment, and I won't lie, I stole this from my favorite professors in the past, and I actually had to find an updated one because the old one was getting real pixelated. An experiment in generating mice. So you take a vase or some big container, and this kills me that they updated it in this way. So it says sweaty underwear. It used to say soiled underwear. Whatever. Somebody's overly fussy. But anyways, you put soiled underwear in there, you add wheat, and you give it the better part of a month. And holy cow, it appears a bunch of mice. They thought these were the key ingredients to making mice. We know that they're getting in there, they're eating the wheat, and they're like, hey, this is a great place for a nest. This debate held until the 1880s. Okay, that's a scary long time for this. Okay, so the story almost ended in the 1600s because this gentleman here, Francisco Redi, devised an experiment to initially disprove abiogenesis. And he did so looking at, again, the example of maggots appearing on meat. So you'll have to let me know if you can see the board okay. I'm wrestling with these new giant overhead screens. So I'll try to do it low enough that it can be seen. Forgot black marker today. Okay. So in Redi's experiment, oh wait, I forgot. I have a diagram for this now. Thank you, OpenStax. They're the first people to come up with a diagram for this, so I applaud them. So he did 3 experiments. Okay, in his first experiment, in his control, positive control here, he has a flask or beaker to which he added meat. And the meat was left open, exposed to the flies. Okay, we know the flies landed, laid eggs, maggots appeared. He says, okay, I believe if you eliminate the flies, you won't get maggots. Okay, he did the same experiment. There's your steak. But this time he added a stopper. Okay, we know flies can't get in, so no maggots. But they argued at the time that air was a key ingredient. And if you cut off the air, They're not going to spontaneously appear. Francisco Redi said, fine. I'm going to repeat this a third time, covering the flask with cheesecloth, right? It's porous. It allows air in, but the flies can't gain entry. So there's your meat, there's your cheesecloth, there's your flies putting or laying eggs on the top instead of on the meat. This last experiment here disproved abiogenesis, right? He showed that it's not the meat that's spontaneously creating a maggot, but the flies are making the maggots. So that should be the end of the lecture, right? We're done? No. There was a problem. And that problem was von Leeuwenhoek's animalcules, right? He had just discovered these new microorganisms, and we did not know how they were reproducing. So, for instance, food spoilage. Okay, you leave chicken broth out long enough, it goes cloudy, turbid, because bacteria grow in it. They thought that food was spoiling because these organisms were spontaneously appearing. So it took 200 more years, it's the 1880s, to figure out that the bacteria, the microbes, were reproducing and not spontaneously appearing. All right, so there's a number of players during these 200 years that were tackling this debate, trying to figure out how to solve it. So, for instance, John Needham versus Lazzaro Spallanzani. They set up almost identical experiments, trying to prove the debate one way or the other. So in their experiments, this is what I thought I was drawing a couple minutes ago. That's not bad. They set up they set up flasks containing chicken broth. Okay, and we knew if you cooked your food, it sterilized it for a time. That's fire. I'm sorry. I can't draw. So, in both experiments, they cooked their chicken broth. Okay? So, in John Needham's version, he cooked the chicken broth, he let it cool. Okay, that's the key part here. They let it cool. And then he put in a stopper, kind of like Redi did. Well, problem is, his chicken broth eventually went cloudy. It spoiled. And he said, well, look, I cooked it, I stoppered it, and it went bad. Clearly, they're spontaneously appearing. Spallanzani saw the flaw in the experiment. He repeated the same thing 20 years later, with one difference. He cooked it, but instead of letting it cool, he stoppered it right away. So he sterilized it, corked it. Spallanzani had the notion that maybe it's the air that's contaminated. Because in Spallanzani's flasks, they didn't go bad. They didn't spoil. Okay, because we know now the air wasn't getting in contaminating it. But, again, they argued air was a vital force. If you stopper it too soon, like with Redi's experiment, they're going to say, you're leaving out a key ingredient. You need that air for them to appear. So, we go 100 years later. We go to the 1880s. Okay, or sorry, 1860s to start. And that's when we get to Louis Pasteur and his very famous experiment that settled this debate. So, in Pasteur's experiments to refute spontaneous generation, two things are going to be important to realize here. One, he is finally going to show that air is gross. Okay, air is filthy, dirty. It is contaminated with microbes. And the other thing is, we were correct in assuming that if you cook the chicken broth, it's going to be sterilized. So in his experiment, you need to cook the chicken broth to sterilize it and still allow air without contamination. Okay, so how do you do that? Well, you cook up a flask nobody uses anymore. a swan neck flask or a gooseneck flask. So, famous Robert Town painting. Here is Louis Pasteur with his crazy swan neck flask. Okay, and you can see in this flask, it's crystal clear. And one where he's broken off the neck has become contaminated. And then here's his wife wondering what he's doing. Okay, so here's the experiment again. The flask begins like this. Okay, long, thin neck. You add your chicken broth. So how does it become bent like this? Well, I've done this myself with other glassware, but in a lab setting, you'll often work with really thin glassware. You heat it with a Bunsen burner and you can mold it. So he heated the neck and he bent it into this ridiculous downward S shape. And then he cooked the chicken broth. And over 100 years later, those flasks are still crystal clear clean. So, sterilized chicken broth, air can still get in, but they didn't go bad. How? Well, if you ever get the chance to see these, I don't know where they keep them, but I've seen other ones that aren't as old. If you ever see one, look right there. You'll see a dirty mark at the bottom of that first bend. Because here's the thing. Okay, at home, we dust. And a week later, we dust again. Much of that dust is microbes. Okay, they fall just like we do, but at a much slower rate. So as the air entered the flask, the microbes fell back down into this first bend, like a trap. So air gets in, microbes get trapped, flask stays clean. So he tackled the problem that Spallanzani couldn't. He cooked the chicken broth, but still allowed air without contamination. And to really hammer home the point, when he did these, he would occasionally bust off the neck on one, and it would get cloudy. So he showed that it was the air's fault. The air was contaminated. So, the important parts that we get out of this. Okay, Pasteur's swan neck flask demonstrated that microbes are, word of the day from last time, ubiquitous. Right? They're everywhere. They're on the ground. They're in our water. They're in our air. They're in and on us. Okay. He also hammered home the idea that, yes, we're on the right track. Fire does kill microbes. Okay. Heat is required for sterilization. Okay, so that led to aseptic technique. So if you already had lab today, you did this, but at 2, if you haven't, we're going to learn that we have to sterilize our equipment with fire when we transfer bacteria. And then last but not least, this proved biogenesis for bacteria. It showed that the microbes were reproducing in some way. They weren't just spontaneously appearing, right? So, one little exception here. But before I talk about this, questions about Pasteur's experiments. Okay, do you understand how that swan neck flask worked? Okay. So once in a while, one of Pasteur's flasks, when somebody repeated it, would go bad. And that was a head scratcher, right? Like, why did it go bad? What did I do wrong? Well, John Tyndall, who was actually a physicist, figured it out. He discovered the presence of what we call endospores. Now, we're going to talk about endospores in lab in a couple of weeks and in lecture. Endospores are only made by a couple of groups of bacteria. But think of them this way. An endospore is like an escape pod. They take everything important in the cell, cram it into a really tough shell, and then they get the hell out of there. And then they just wait until conditions become good again. What Tyndall found out is that these endospores were surviving the boiling. They were just that tough. So this kind of put to rest everything with this debate. And one of the other things that was interesting he sorted out is that different endospores have different levels of toughness. Okay, so for instance, in lab today, We worked with Bacillus subtilis, or you're going to work with it. B sub makes endospores, and they can last a good 14 minutes of boiling. That's long enough to make mac and cheese. That's a long time. Clostridium perfringens, a lot of pathogenic Clostridia. That guy causes gas gangrene. That's a good 20 minutes of boiling can survive. And one that's just bananas, Clostridium botulinum, botulism. Okay, 360 minutes of straight boiling. This guy used to be the bane of the canning industry. Okay, now we know you can kill it with pressure and heat, so it's not really much of an issue any longer. Okay, almost through the good historical stuff. So the next thing we're going to discuss, briefly, are some of the players involved in germ theory. Okay, so what's germ theory? Germ theory is the theory that diseases can be caused by microbes. So cholera is caused by Vibrio cholerae. Enterohemorrhagic fever can be caused by certain types of E. coli. Chickenpox is caused by varicella zoster. Prior to germ theory, we had no clue what caused disease. Bad luck, a curse, okay, voodoo, whatever. So the first person to start getting a notion of this was Joseph Lister. So he was a surgeon in the mid-1800s, I think Civil War, and he got this crazy idea. If I disinfect my equipment between patients, they're gonna live longer. And that's exactly what he did. He was the first surgeon to really disinfect their equipment properly. And he did so using phenol. Okay, concentrated phenol is horrible stuff. Okay, these days you really only see it in floor cleaner, like pine sol. But at the time, it was great for cleaning surgical equipment. And when he did so, the patients didn't get sick. So we thought, well, maybe there's something on the equipment. Maybe it's one of these anti-molecules. So 1876, not long thereafter, you had Robert Koch or Cook or whatever. Robert Koch is the father of germ theory. He is the one that figured out that a disease can be caused by a specific microbe. So how did he do that? He devised a series of postulates, fairly brutal experimental steps. Okay, so we go through this, keep in mind, 1800s. So let's use anthrax as an example. Okay, people get anthrax, cows can get anthrax. So imagine you have a farm with cows, and some of the cows are showing blisters or pustules where the center of that blister is becoming necrotic, turning black. Okay, so some of the cows have the signs of anthrax, other cows are still healthy. So that's the first postulate. Some of the animals are sick, some of them are healthy. Second postulate, and this is fun. You go to one of those sick cows with a swab and you go to swab one of those blisters. And then you just put it out on a Petri plate like we did the other day. You want to try to isolate that suspect pathogen. You want a pure culture. If you get that pure culture, you go on to step 3. Sorry for this. You take cells from that plate and you use it to make some healthy cows sick. So you inoculate the healthy cows and you wait. And you wait and you wait and you wait to see if they show those same blisters. Okay, if the newly infected cows show the same signs and symptoms, you go to step 4. Swab again on the newly sick cows, re-isolate. If it's the same pathogen on that second plate, congratulations. You just isolated and potentially identified your pathogen. Okay, this is how he figured out that anthrax is caused by a microbe, by Bacillus anthracis, a actually wickedly common soil microbe. And then later on in mice, he demonstrated that tuberculosis is caused by Mycobacterium tuberculosis. Now that one's really impressive Because unlike most bacteria that we work with, where you wait 24 hours and they're grown up and ready to go, tuberculosis cells take a week to 2 weeks before they show any signs of growth. So this dude had impressive patience. And these are old postulates, okay? There's more modern postulates today that allow us to identify what pathogen is causing what disease. All right, but any questions with germ theory before I hit my last story here? Okay. So the last story I have here is the story behind vaccination. Okay, and it's actually a really old story. The first vaccine was administered longer ago than you'd think. So the first disease we attempted to vaccinate against was smallpox. Okay, you don't hear about smallpox much anymore, and you're going to find out why. But smallpox is like chickenpox on steroids. Okay, you don't just get kind of generalized covering of rash and blisters. The entirety of your skin becomes covered in these nasty blisters. There's a lot of complications. Mortality rate was very high. And I read about cases where the blisters would become so severe that the dermis would separate from the muscular tissue. If an individual survived, 2 things happened. 1, you were scarred for life. 2, you were immune to smallpox. So, if you survive smallpox, you could never get it again. So keep that in mind. So, the person who was studying this, trying to deal with it, was Edward Jenner. Okay, and knowing that you could survive smallpox and never get it again, he hypothesized that if you were exposed to a less virulent pathogen, okay, something not as bad but similar to it, you could get immunity to the more virulent pathogen. So, if you survive its weaker cousin, you get the same immunity. And that's actually an old idea. Okay, probably for, I don't know, at least 1,000 years, the Chinese have been using variolation, where they'd purposefully give individuals tiny doses of smallpox to try to give them milder cases. Okay, so Jenner wanted to do a twist on that. Instead of giving it the same pathogen, you give the patient something similar but not as strong. So he did a lot of research. He traveled the world trying to find a good cure, and that brings us to a dairy farm. Heard a story about some milkmaids that said they couldn't get smallpox, but they never had it. So he went, he talked to them, he investigated, and what he found out was that the milkmaids had survived cowpox. There's a number of different pox viruses. They're all in the same family. They tend to be species specific. Okay, cowpox, monkeypox, camelpox, whatever. Okay, smallpox is human pox. So with cowpox, an individual can get it. This is an absolutely ridiculous case. Usually somebody will get maybe like a couple of these blisters, but it's extremely mild. And when you get over cowpox, you can't get smallpox because they're so closely related. So Jenner's like, well, bingo, this is what I've been looking for. I'm going to give people cowpox virus so that they won't ever get smallpox. And that's exactly what he did. So folks, 1796, May 14th, if you want to be really accurate, he administered the first smallpox vaccine, which was just injecting that woman with cowpox particles. Now, what happens is you get a big angry blister at the point of injection. It eventually goes away, and you're stuck with a scar and immunity. Okay? Being the 1700s, early 1800s, this didn't fly with a lot of folks. Okay? They were scared of getting this cow disease, and they thought they'd grow tiny cows from their appendages. But folks got on board because starting in the 1960s, the World Health Organization said, you know what? We're done with this. We're going to work together, and we're going to eliminate the first disease from the face of the planet. Okay? There's a reason you don't hear about smallpox anymore. So over about a 20-year period, ending in the 1980s, the World Health Organization went around, vaccinated everybody who'd sit still, did mass quarantines, and they eradicated smallpox from the face of the planet. With one caveat. 1980s equals Cold War. So, the US and the USSR agreed upon keeping their own samples. But it wasn't whole virus. It was supposed to be just genetic material. So, in Atlanta, Georgia, the CDC facility, there is smallpox genetic material. Was Vector Labs in the USSR, they kept theirs as well. And that was the end of it. With one exception. About 4 years ago or so, it was in the news that somebody was cleaning old closets at the CDC and they found vials of smallpox. So I'm hoping that was the end of it. But that was a pretty impressive task, right? Working together to eliminate an entire virus from the face of the planet. And it's a notion we still use today. So in Southeast Asia, a lot of folks don't realize we are this close to eliminating the guinea worm. Okay? Every other year we think it's gone, a couple more cases pop up, but it's almost dead. And then the Bill Gates Foundation, they are investing insane amounts of money to eradicate polio. So I think that one will be gone in our lifetimes. But she's pulling a guinea worm out of her foot. But the rest of this, I'll be brief. I just like to end the first lecture with showing you folks, this class is a stepping stone. Okay, it's a stepping stone to many other disciplines. You can go into bacteriology, where you just study bacteria as a career. If you're really hardcore, you go and be a mycologist. There's not a lot of money there anymore, but you study fungi. Parasitology, good luck. You go and study protozoan parasites or helminthic worms. Immunology. Immunology is the part of micro that I always dreaded because everybody's got something they're not great at. Immuno was the thing I wasn't great at. But your immune system and its interaction with pathogens is a part of microbiology. And this is actually a really good class to take in the future, virology. And virology is good because every chapter is the same thing with just variations. So it's pretty easy.